The computer scientist or statistician who has the expertise to use advanced analytical techniques usually lacks the biological background necessary to understand biological phenomenon. In this week's module, I will provide an overview of relevant biology background that underlies genomic assays. Specifically, I will describe cells, DNA, including the central dogma of molecular biology, a brief overview of Mendelian genetics, and that will provide the necessary background to then introduce hybridization assays, which are used in genomic studies. First, we will discuss cells. Cells are the fundamental units of all living organisms. Each cell is a complex system consisting of many substructures. There are two main types of organisms. The first, prokaryotic, are unicellular organisms, for example, bacteria, and lack a well-defined nuclear membrane. The second, eukaryotic, are multicellular organisms having a nucleus, and examples include humans, dogs, and plants. In this course, we will focus on eukaryotic cells. The nucleus is a subcompartment found only in eukaryotic cells, in which the organism's DNA resides. Enclosing the nucleus is the nuclear membrane, the protective wall that separates the nucleus from the rest of the cell, which is called the cytoplasm. The entire cell is enclosed by the plasma membrane. Embedded within this membrane is a variety of protein structures that acts as channels and pumps to control movement into and out of the cell. Here are two ideograms depicting a eukaryotic cell. On the left, you see the different organelles within a cell. On the right, it attempts to illustrate the three-dimensional nature of a cell. The nucleoplasm includes everything that is in the nucleus, and the nucleus takes up about 10% of the volume of a cell. The cytoplasm includes everything in the cell except the nucleus, such as organelles, water, and other chemical molecules. Organelles are specialized subcellular structures in the cytoplasm where each has a very specific function. For example, ribosomes are complexes of RNA and proteins whose main role is to translate messenger RNA into chains of polypeptides using amino acids delivered by the transfer RNAs. Next, please watch this short YouTube video which further explains the structure of cells.